What is accommodation reflex? Before we get to the accommodation reflex and understand how the lens work, I just want to give you a reminder of the difference between the two types of lenses. We have a convex lens and a concave lens. When I have a concave lens, this lens is actually when the light comes in, when the light comes in to that lens, it acts as diverging a medium for the rays, for the light rays. All right. So the concave, it brings the rays, they come to the lens itself and then they become divergent. But the concave, it brings all these rays and becomes convergent. Okay, so it converge these rays into a focal point. Why is that important? It's important for you to understand that the vision, if you have a near vision and if you have a far vision. I will explain to you the concept, but afterwards I will give you keywords on how to answer these questions in the exam without even thinking about the explanation itself. But I have to explain to you the idea first, and then I will tell you how to answer these questions quite fast. Now, what happens to the lens if we have something that's near to us, and what happens if we have something that's far to us? The idea here is, in order to accommodate for the light and for the object that I'm looking at, I need the function of the ciliary muscle and the iris. These two are going to help me to modify and to accommodate to the object I'm looking at. Now, what will happen? First of all, I have the lens. Normally, in normal circumstances, the normal circumstances is that the suspensory ligament that are attaching the lens to the ciliary body is taut. It's actually active. It's all the time active. So it's making my lens less puffier. All right? This is because this is my normal circumstances. I'm looking at a long vision most of the time. However, if I bring a book near to my eye, I need to focus my vision. I need to bring, remember when we talked about the convex lens, we said it brings it to the focal point. I need to focus because if something comes near to my eye, my rays are going to go far away. So I want to bring them to a focal point and I want them to fall on the retina itself. Normally, if I look at something far away, because it's very far, as they reach to my lens, they are coming parallel to each other. They are already parallel. So it's very easy for them to fall on the retina. But if I have something that's near to me, they don't come parallel. They come away from each other. So I really need to make my lens buffy. I need to collect all these rays to a focal point so that it can fall on my retina and I can see. Now let's get back to that. If you think about your lens, it's normally taut, it's already stretched by the suspensory ligament. This is the normal vision if I look at things away from me. Now, if I have a book or something that's near to my eye, I need to make my lens puffier. So the ciliary muscles are going to contract. If the ciliary muscles contract, the suspensory ligaments are going to lax. They are going to become slack. If they become slack, the tension on the lens is going to be removed. I don't have tension on the lens, so the lens becomes puffier. So if I have it puffier, because the object is very near to me, that will help me to bring all these rays into a focal point so it falls in the retina so I can see. All right, so remember that this is actually happened by the parasympathetic innervation through the short ciliary nerve. So if I have a near object, I need to activate my accommodation reflex. To do the accommodation reflex, it's for the near objects. It's for the short vision. For the short vision, my lens is going to become puffier. The muscles are going to contract. The ciliary muscles are going to contract. The suspensory ligaments are going to lax. It becomes puffier. Now I can bring all these rays into a focal point on my retina so I can see nicely. So what's the magic letter we have here? It's the R letter. So for short vision, parasympathetic innervation through short ciliary nerve, that's going to cause contraction on my ciliary muscles, and that is going to make my lens puffier. As an advice from me, remember it as R letter, memorize only one thing, and in the exam, if you are asked about the opposite thing, you know that this is the opposite of the information you know. So what you need to have in your mind now, the R letter for the short vision, puffy lens, ciliary muscles contracted by parasympathetic innervation, it reaches contraction of ciliary muscles, it reaches by short ciliary nerves. R, 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 puffy, short vision. If you have a question in the exam about a long vision, it means that the lens is tensed, the short muscles are relaxed, all right? And that is by sympathetic innervation, by long ciliary nerve, and some contribution from the short ciliary nerve, all right? Now, what happens if we have the suspensory ligament taut already? It means the lens is already stretched. But if I have contraction of ciliary muscle, if they contract, they will relax the suspensory ligament and the lens is going to become puffier. Okay, so please memorize only one thing and the other thing will become very easy. For short vision, puffy lens, ciliary muscles contracted, the suspensory ligament is the enemy of the ciliary muscle, so they will become relaxed. That will be by parasympathetic innervation, and these neurons or fibers are carried by short ciliary nerve, and this is for short vision. And the opposite is true for the long vision. Now, what happens to the iris then? This here, we explained everything related to the ciliary muscle. What about the iris? Remember, you guys, the iris was your diaphragm. And because your light, it goes into the eyeball through the defect in the iris. I have the pupil. My pupil is what brings the light to my eyeball. What will happen if I have something that's near to my eye in a near vision? Do I really need to have a lot of light rays coming into my eyeball? Of course, no. So I want to reduce the amount of light coming into my eyeball because that structure is already near to my eye. So what will happen to my iris muscles? The sphincter pupillae muscles are going to contract because they want to reduce the amount of light coming from this very near object. So again, here we are, R letter, the parasympathetic innervation by short ciliary nerve for the short vision are going to activate and cause contraction of the sphincter pupillae muscle. All right, sphincter, not dilator. Okay, so the sphincter or constrictor, constriction, not dilation. Constriction has the letter R, constriction of the pupil. All right, not the dilator pupillae. All right, so memorize only one thing and the other one will be the opposite to it. For the short vision, you need to activate those muscles and constrict your opening because you really don't need all of these things. So you are acting on both ways. You are working on the lens itself by making it puffier by the activation of the ciliary muscles, all right? And we are also acting on the iris by activation of the sphincter pupillae, and because they are arranged in circular manner, if they contract, they will constrict the pupil. However, the dilator pupillae muscle, they are arranged in radial manner, or how can we say it, in what, radial. Yeah, so if they contract, they will dilate the pupil. So unfortunately my pencil is not working, so I cannot, but I think this the image is good enough to tell you that the sphincter pupillae, it constricts because they are circular. If they contract, they constrict the pupil, and the radial ones, if they contract, they bring it far away and it becomes the pupil becomes wider, so you get more light because you want to see the very far object. All right, Now, what's the histology of the iris? Now, think about your iris. It's basically, you guys, your iris is very important because this is what gives the color of your eye. For example, if you look at, do I have it here? I'm not sure. Okay, so anyway, I'm sure, yeah, I had it previous. This is, can you guys see this brown eye? Because this is the melanocytes in the stroma of the iris, all right? So the iris is what basically gives you your eye color. So it based on the amount of melanocytes you have them in the stroma, you get to have your eye color. And down there you have the pigmented layer, because we need to have pigmented layer. And in the middle you have your muscles. If you have muscles that are near to the pupil, okay, you guys know that the pupil is here, the circle you have, this is going to be the sphincter pupillae. If you have muscles far away, this is going to be the dilator pupillae, because as I said previously, if I have muscles near to the pupil here, this is your pupil, this is going to be the sphincter pupillae. If you have them far away, radially arranged, it's going to be the dilator pupillae. The same thing you apply to the histology. If you have, this is your iris here, this is the space of the pupil. If you have a muscle that is near to the pupil, this is going to be the sphincter pupillae. If you have it far away, and this is the pink part, it's your muscle, it's going to be the dilator pupillae. And this is the structure of the lens. It has all the protein crystalline. It has all these lens fibers. As the lens fiber muscles, cells, they mature and they lay here to form the lens. They are going to lose their nuclei, so they look like a fiber, but actually they are not fibers. They are cells that lost their content, and that protein is actually forming the core of your lens. And if you look at it's basically, you have the sclera, and here you have the ciliary muscle, and you guys know that the ciliary body itself, it has the muscle, and it has the process that forms the aqueous humorous. And here is your iris. All right, so this is your iris, this is your ciliary body, it has ciliary muscle and ciliary process, and from here you're forming the aqueous humorous. And when you bring it from the posterior chamber to the anterior chamber, you are bringing it to the sclerovenous sinus. All right. So if you bring it, the aqueous humorous, all around the iris, and you bring it to the scleral venous sinus, you have it in the canal of Schlemm. It gets absorbed, and then you have it in the scleral venous sinus. Now, this is a recap to show you the three layers of the eyeball. We already talked about them. The sclera, and followed by the sclera, the vascular coat, and then your retina. The retina has a neural one, non-photosensitive and photosensitive part. And outside you have your pigmented layer, because you want to absorb the light. Now, what is the corneal reflex? Corneal reflex it's basically, reflex means you have an afferent limb that brings information. You have interconnection between two nuclei, one from the sensation you're getting and one to the motor innervation that's going to have your efferent limb. So if you touch here, it means you are activating the ophthalmic nerve, because you know that the sensation is carried by trigeminal nerve from my eye by ophthalmic division. So I have an afferent limb. As I touch, I have afferent limb, and that limb is coming all the way to the trigeminal sensory nucleus. From there, you are having your connection to the nuclei of the seventh cranial nerve, and that seventh cranial nerve will give you the efferent limb, and the orbicularis oculi muscle is going to contract and makes you to close your eye. Okay, so this is what we mean by corneal reflex. And what about the retina? Retina, now here is your vision. As you get your light inside and you want to see, you have so many layers in the retina, and you need to process the information all the way until you get the information through the optic nerve and you can see. Now let's see, how do we actually see? But before that, I want you to generally know that in the eyeball, by the ophthalmoscopy, you get to see the eyeball. It's important for you to know that you have an area of your central retinal artery and veins and everything that gives you all this information. It comes in with the optic nerve. So the optic disc is here. This is the place where you are forming the optic nerve and it comes out of the eyeball. There is a very important area in your eyeball called the macula lutea. Macula lutea means it's a yellow spot. In that yellow spot, you have a depression called fovea centralis. Why this is the central fovea is important? It's an area of the central vision. This is the area that you have the very high sensitivity to light, because in that area you have a huge amount of cones, and cones are the photoreceptors responsible for the colored light. I think I have a diagram. Not Okay, so here we have the highest amount of cones. It's very sensitive in bright light because cones are the photoreceptors that are responsible for the colored vision, and the rods are working in dim light. So in this spot, it's the central vision. If you have a problem, I will show you a picture on how that patient will see. And this is the importance of the fovea centralis, all right? And what would happen in the retinopathy? So many things can happen. Like this is an example of the retinopathy. It means that there is a problem going on with the retina. That problem could be by a hemorrhage in some of the capillaries of your eyeball from the branches of the central retinal arteries. You can have your cotton wool appearance, which means there is occlusion of the central artery or any of its branches. You could have microaneurysms. You can have abnormal problems with the blood vessels. And if they are left untreated, they will eventually end up with blindness. All right, so this is how the retinopathy would look like under the ophthalmoscopy of that patient. You guys know that the optic nerve here is at the optic disc. You know that the macula lutea and the fovea centralis is in the center. This is important for the central vision. But if you have a patient with retinopathy, you will find all of these appearance, the cotton wool spots. It means there is occlusion of the branches of the central retinal artery. There could be anything going wrong, collection of oxidates, and neovascularization and problems with the capillaries of the central retinal artery or its branches. All right, and that can eventually affect the vision. Now, the question is, how do we see? Now, you guys know that the light will come in, it goes through the cornea, and it has a refractive index, that the highest refractive index is actually in the cornea. And as you proceed, refraction is important because the light wants to move on from a medium to another. Here, from the air to the aqueous humorous, and then from there to the lens, and then from there to the vitreous humorous, and then it falls on the retina. Now, if you guys look at the retinal layers, you have the layers from outside to inside. What I want you to understand is, I know that this is the bottom of the eyeball and this is the most posterior structure. It's the pigmented layer and the choroid. But the idea is as follows. As the light comes in, it hits the internal layers of the retina without doing anything. The light keeps going, it keeps going, it keeps going until it hits the pigmented layer. At that point, the light photons are going to get absorbed and they are going to get processed by the layers of retina retrograde again, back all the way up until they are getting all the way by the ganglion cells that are forming your optic nerve. And from the optic nerve, you take it again to the brain. So again, as the light comes in, it hits all the layer or the internal layer of retina without doing anything up until they reach the deepest layer, which is the pigmented retina. And from there, it gets processed back from the back to the front, all the way until they reach the innermost layer of the retina, and then it gets processed through the optic nerve all the way to the brain. So what are these layers? You have it coming, and why this is the reason actually why this happens. As the light comes in, these cells are neural cells. They don't understand what light means. So the light keeps going until they reach the photosensitive cells, the photoreceptors, the cells that has the ability to transduce the energy from the light energy into electrical energy that my brain can understand. In that case only, I will process this information. I'm going to transform the photons of light into electrical energy, and I take it all the way retrograde back side again through the neurons of the horizontal cells, and from there to the bipolar cells. And you guys know the bipolar cells are important for the sense organs. And then from there to the amacrine cells, from there to the ganglion cells. And these are actually the cell bodies of the optic nerve. So if you have an exam question about which of the following cells in the retina are the cell bodies of the optic nerve, it's actually the ganglion cell layer, because these are the cell bodies of the optic nerve. And from here, you get the information all the way through the optic nerve. You get it processed in the brain up until you reach the visual cortex. If you guys look at the retina, you have all of these layers, all right? You have them from the outside to the inside as the outer lemmatic membrane. You have the outer nuclear layer. Why do we call it nuclear? Because it has nuclei of these cell bodies, of these neurons. What does outer plexiform layer mean? It means plexiform in Latin means you have nerves, you have wires. So these are the axons and dendrites of the neighboring neurons. And this is inner nuclear because, again, this is the next group of cell bodies. And because you know that the collection of cell bodies in the central nervous system, we call it nuclei. So these are the nuclear layer. And then from there to the inner plexiform layer, and from there to the ganglion cell layer, and then all the way to the optic nerve. So once you have the cell bodies of the optic nerve, you get it all the way by the central processes of these ganglion cells, take it all the way, and you form your optic nerve. Now back to this, if you look at a place where you only have the optic nerve and you don't have the retinal layers, this is what we call the optic disc, because at the optic disc, this is what we call a blind spot. I don't have retina here. This part, it does not see, it's not photosensitive, it cannot do anything because I don't have all of these layers that are capable of transforming the photons of light into electrical impulses. All right, so if you guys look at these retinal layers, you don't have them, as the axons of the ganglion cell layers are going to get deep inside by your eyeball and taking the information to the brain. So the optic disc is the blind spot. You have the axons of the optic nerve. You don't have the retinal layers that will help you to see. So this is a blind spot in your eye ball. All right. And this is your lens. All right. You have the stroma, you have a Bowman's membrane, and you have the Dissemin membrane. You have epithelium outside and endothelium inside. So it's very simple to know the layers of the lens. How will you remember the names of the membranes? Remember that toward the epithelium, B letter comes first. So Bowman's membrane is outside near to the epithelium. Dissemin's membrane is inside near to the endothelium. And other than that, you have your stroma. This is all the fibers, the lens fibers, and I talked about the mature lens fibers previously. What will happen if we have collection of pigments in the lens? You will have reduction in the transparency of the lens. There will be opacity of the lens, and this is how it looks like. It's totally different than the glaucoma, because the glaucoma is everywhere in front of the iris. It covers the whole eye behind the cornea. But if you have a problem with the lens, then it's only concise to the lens. And there are also other investigation on how to differentiate between them. The last thing we have here, guys, is the visual pathway. Now, how do we process the information of light, and how can we differentiate between the diseases of the vision? This is the ciliary ganglia. I already talked about the ciliary ganglia. Before we talk about the visual problems, let me tell you the last thing here is the ciliary ganglion. Remember when I told you that the parasympathetic innervation, it goes through the short ciliary nerve. So you get the information from the Edinger-Westphal nucleus, from the oculomotor nerve, from the brainstem, and it synapses here with the postsynaptic neurons, and after it relays in the ciliary ganglion, it goes by the short ciliary nerve to your sphincter pupillae and ciliary muscle. However, the sympathetic fibers, which is coming from the superior cervical ganglion, from the spinal cord, it goes through the ganglion without synapsing. Some of them can go through the short ciliary, some of them they can go through the long ciliary. They only go to the dilator pupillae muscle, but they don't go to the ciliary muscle, because normally the suspensory ligaments are already acting on the lens and they are making the lens flattened for long vision. So remember, guys, if I have accommodation reflex, I have it by the short ciliary nerve, R letter. It goes to the ciliary muscle to contract, to make the lens puffier. And it goes also to the sphincter pupillae, because I want to reduce the light coming into my eye. However, for long vision, it has nothing to do with the ciliary muscles, because the suspensory ligaments are already making the lens less puffy, but the sympathetic is going to act on the dilator pupillae to dilate the pupil in the iris. All right, so we have two things in terms of short vision, only one thing in long vision. In short vision, contraction of sphincter pupillae by parasympathetic innervation, and contraction of ciliary muscles, because I want to encounter the suspensory ligaments. However, for long vision, it's only for the dilator pupillae. The ciliary muscles are already inactive. The suspensory ligament are already making my lens less buffy in normal circumstances. So short vision, two things. Long vision, only one thing. Ciliary muscle, it acts only in parasympathetic innervation in the short vision. However, it's already relaxed. Okay, now last thing is the visual pathway. I will stop and make it in a separate video because my Apple Pencil is not working and I really need to write it down. Thank you guys for watching. I will make another video for this last concept. I hope you grasp the concept and I wish you all the best.